Hello and welcome to this lesson for Advanced Higher Biology, which is a continuation of the previous one where we looked at the generation of a nerve impulse, and this is essentially the process by which this would happen if we had an environmental stimulus, and we're going to use the vertebrate eye as our example. So what you're expected to be able to do is understand the nerve response due to an environmental cue. And you need to be able to name the cells, proteins and protein complexes found in the vertebrate eye. You need to name and describe the actions of the proteins involved in the reaction cascade. You need to explain the role of ion channels in affecting the neurons, and you need to be able to describe how this process is slightly different in the cone cells of the vertebrate eye. So nerve impulses can be generated in response to an environmental stimulus, and a very good example of this is in the vertebrate eye, where light is detected by two different types of cell, which are found in the retina. And the retina is the tissue at the back of the eye, this blue section along here. So light is entering through the front of the eye, through the cornea and the pupil and the lens, and it's being delivered to the back of the eye where it's detected by the cells which are present in the retina. These cells are then going to transmit a nerve impulse to be transmitted to the brain. So it's essentially the job of these cells within the retina to convert that light energy into electrical energy and move it to the brain where it can be processed. So these cells specifically are rod cells and cone cells. Within the retinal tissue, we've got many more rod cells than we do cone cells, around 125 million to 6 million. So 125 million rods to 6 million cones in the human retina. Rod cells, which are these ones here, are sensitive to changes in light intensity. So they are useful for vision in areas of low light intensity, for example in a dim room. And nocturnal animals have a greater proportion of rod cells, so they therefore have a better vision at night. Cone cells, on the other hand, these ones here, are not as sensitive to light as rod cells. However, what they are sensitive to is specific colours or wavelengths of light, so green, red and blue, and in some animals also UV light. And it's these cone cells that allow animals to have colour vision. Now, as you can probably guess, this is going to involve some kind of protein. So the protein involved in this detection of light is a protein called rhodopsin. And this comes from a light-sensitive molecule called retinol. Retinol is involved in capturing the light energy. It is a prosthetic group which is covalently bound to a membrane protein, so protein that's embedded within the membrane. We have retinol covalently bound onto it, and this membrane protein on its own is referred to as opsin. So retinol and opsin together form the retinol-opsin complex, referred to as rhodopsin. And it's embedded within the membranes of the photoreceptor cells in both rod and in cone cells. Now, when this protein is exposed to light, it's going to set off a cascade, a signalling cascade, which will result in the transmission of a nerve impulse. Essentially what's happening is that retinol, which is bound onto the opsin, forming rhodopsin, is going to absorb a photon of light, and that will cause a conformational change in that rhodopsin, changing it to photoexcited rhodopsin. Photoexcited rhodopsin therefore activates a G protein, and if you remember when we talked about signalling molecules previously, we noted that one of the cascade options was the G protein cascade. So in this case, it activates a G protein which is called transducin. And transducin can actually be activated as more than one molecule at this point. So one single photoexcited rhodopsin is able to activate hundreds of molecules of transducin. So this one individual molecule here in this diagram, it is activating this one. However, once one has been activated and moves on, it's able to activate more. So over time we get hundreds activated just by this single photoexcited rhodopsin molecule here. Once our transducin has been activated, It therefore activates an enzyme, which is this one here, this PDE enzyme, so phosphodiesterase. So transducin is going to be activating the PDE enzyme, and again, this is slightly different. We've got our transducin molecule, is each single transducin molecule can activate one single molecule of PDE. However, if we've got hundreds of these activated, therefore they can go off and activate hundreds of molecules of PDE. So we've gone from transducin to activating our PDE. PDE heads over towards our cell membrane, and its job is to catalyze the hydrolysis of a molecule called cyclic GMP or cGMP. And each active PDE molecule is able to break down thousands of cGMP molecules per second. So we've gone from activation of rhodopsin by our photon of light. That's created photoexcited rhodopsin. That photoexcited rhodopsin is able to activate several transducin molecules, so hundreds of transducin molecules have been activated. Each one of those activates an enzyme called PDE or phosphodiesterase, and that phosphodiesterase molecule can then come over and catalyze the hydrolysis of a molecule called cGMP or cyclic GMP. And each one of these molecules is able to catalyze the hydrolysis of thousands of GMP molecules. This reduction of this cGMP to just GMP, therefore, causes eventual closure of ion channels in the membrane of rod cells. So normally in darkness we have these open ion channels. In this case it's a sodium ion channel, so it's usually positive ions that are coming in, leaking inwards, so things like sodium, calcium are normally leaking in, and that's obviously going to affect the polarization of the membrane, preventing it from going into hyperpolarization and producing a nerve impulse. When this PDE molecule is able to come over and it catalyzes this hydrolysis of this membrane, of this molecule, cGMP, once it's been hydrolyzed and turns into GMP, that as a result is going to close the channels, or ion channels down here. And as a result, we can therefore lose this leakage inwards of these positive channels, and this generally changes our membrane potential. So the membrane potential increases, we have an increasing charge or hyperpolarization, and that is going to trigger our nerve impulses in the neurons in the retina in the same way that we've already seen. So this protein cascade is able to provide a high degree of amplification. So we have stimulation by a single proton, resulting in a large effect on ion movement across the membrane. That means rod cells have a high sensitivity at low light intensities, and rods can generate a nerve impulse from as few as 10 photons of light. So simply having 10 photons of light is able to eventually catalyze the hydrolysis of thousands and thousands of these molecules, closing these ion channels, allowing for hyperpolarization or the increasing charge of the membrane, and triggering therefore a nerve impulse in the neuron in the retina, just in the same way as what we've already looked at. This process is slightly different in cone cells, as we're not responding just to light, we're responding to colour. So in cone cells we have different forms of opsin, and they combine with retinol to give different photoreceptor proteins. So they're not forming rhodopsin, they're forming some different photoreceptor proteins. Each one of these has a maximal sensitivity to specific wavelengths. So they generate a nerve impulse in exactly the same way as rhodopsin does in rod cells, but in response to a specific colour of light. So we have one photoreceptor molecule which is able to absorb blue, one for green, and one for red. Each individual cone cells only contains one type of photoreceptor molecule, so when that is activated by its particular colour of light, that sets off the same process as what we've just seen in rod cells, but it allows us to detect these individual colours. So you should be able to name the cells, proteins and protein complexes found in the vertebrate eye in that we have both rod and cone cells, and we've got retinal and opsin forming the retinal opsin complex called rhodopsin. The name and describe the actions of proteins involved in the reaction cascade, where we've got rhodopsin changing conformation to photoexcited rhodopsin, activating a G protein which is called transducin. That transducin molecule activates our enzyme phosphodiesterase, or PDE. That PDE catalyzes the hydrolysis of a molecule called cyclic GMP, or cGMP, and as a result we're able to change the closure of ion channels, resulting in the changing in membrane potential and triggering our nerve impulse. So the role of ion channels in affecting the neurons, well, you should know that previously as we change the concentration of ions we're able to change the membrane potential, and changing the membrane potential eventually triggers our wave of polarization along the neuron and allows it to affect the other end of the neuron. So at the axon terminus we may release a vesicle containing neurotransmitters, or stimulating a similar cascade in another neuron. This process is slightly different in cone cells because they are not simply just responding to light. Rather than opsin combining with retinal to produce rhodopsin, it's giving us 3 different photoreceptor proteins, each with a maximal sensitivity to a specific wavelength. Those photoreceptor molecules are detecting blue, red and green light and allows mammals and other eukaryotes to be able to detect vision in colour. As ever, feel free to ask any questions on this as we go over it later on.